In our previous videos on the basic processes of the central dogma, that is transcription followed by translation, we have learnt how the specific sequence of nucleotides it defines the sequence of amino acids in a protein. Now, is this simple chain of amino acids, that is the protein? No, this biomolecule, which performs a range of functions in the cell system, it takes a range of conformations. So, in this video and the next two videos that will follow, we will learn about the structural levels of organization that leads to the formation of the various functional proteins. Now we have seen that the specific order of genetic information which is present in the messenger RNA molecule, it in association with the ribosome and with the help of the adapter molecule, that is the transfer RNA, it leads to the formation of a specific order of amino acids which are joined to each other or linked to each other by means of peptide bonds. This basic structure in which various amino acids they are linked to each other is known as the primary structure or the 1D structure of proteins. Now, this polypeptide molecule, this macromolecule that is formed by the linking of these various amino acids, if its size is more than 50 amino acids, then it will form a protein, or rather it is known as a protein. However, if the number of amino acids is less than 50 amino acids, then the macromolecule formed is known as a peptide. So, the basic unit structure of the primary uh, protein or primary structural level of protein is the amino acid. And let us revise once again that the amino acid, it is basically the organic molecule in which there is a carbon atom having both the amino group as well as the acidic or the carboxyl group. And along with these two functional groups, that is the amino group and the carboxyl group, the acidic group, it has got a side chain, the R, and it is basically this R group present on this alpha carbon that differs or that leads to the various amino acids. Now, at the time of translation on the ribosome, we have seen that the amino acid that was present on the P side, P site of the ribosome, it forms a peptide bond with the incoming amino acid present on the transfer RNA at the A site. Now this we have already talked about in our previous video on the translation. So the peptide bond that is formed between the already existing amino acid and the next amino acid that enters the chain, it is basically, it results from a dehydration synthesis. That is, there's a dehydration synthesis of the already existing or the upstream amino acid and the incoming or the downstream amino acid. the carboxyl group of the amino acid present, it reacts with the amine group of the upcoming amino acid, and a water molecule is removed. Now, this process, that is this dehydration synthesis, which leads to the formation of the peptide bond or the amide group, it is catalyzed by the ribosome itself, a ribozyme catalyzed reaction it is. So a molecule of water is released and it leads to the formation of an amide structure that is known as the peptide bond between the two amino acids. And now this structure, it has got the two terminal regions. One is the N-terminal region and the other is the C-terminal region. And it is this C-terminal region which is available for the formation of the next peptide bond with the next incoming amino acid. So we say that the translation, it proceeds from the N to C direction, because the upcoming amino acid, its N terminal, the amino group, it will react with the carboxyl group of the existing chain. Now, one thing that is obvious over here is that the side chain of these amino acids that are forming this peptide sequence, polypeptide sequence rather, they are in transpositions, because in the entire molecule of amino acid, these R groups, they are the bulky part, they occupy more of space. So the conformation of these bulky groups or the R groups in the adjoining amino acids, it is normally the trans conformation. So in this way, peptide bonds, they are formed between adjoining amino acids. Now we know that in a biological system there are 20 essential amino acids, and the R group of these 20 essential amino acids, they differ in their chemical properties. On the basis of the nature of the R group present, these amino acids, they may be of different types. They may be hydrophobic or the aliphatic amino acids. They may be the aromatic hydrophobic amino acids, or they may be the polar uncharged amino acids which are hydrophilic, or they may be acidic, which again are hydrophilic amino acids. And another category of hydrophilic amino acid is the basic amino acid. So they may be uncharged, they may be acidic, or they may be basic. All these three categories, they belong to the hydrophilic group, whereas the hydrophobic group, it may be aliphatic or aromatic. So these specific groups, the R groups, they define the shape of the mm, protein that is formed, and it is this specific shape of the protein which defines its uh, site of occurrence or the various functions that are carried out by these biomolecules, that is the protein. So this amino acid sequence, which is very specific, Because of the specificity of this amino acid sequence, the protein will take a particular shape, and that shape will define whether it can exist within the lipid membrane, that is the hydrophobic amino acids will be present in contact with the lipid molecules, or if it is present in the cell cytoplasm, the hydrophilic amino acids, they have to be on the outer surface of the protein. So in this manner, these side chains of amino acids, they define the shape of the protein, and depending upon the shape of the protein, the occurrence of its active site, the various functions of the proteins, they are defined. And these side chains, they interact not only with the surrounding environment, but they also interact with other parts of the polypeptide chain or the protein molecule also. So this leads to the folding of the mm, amino acid polypeptide sequence, resulting in the formation of the secondary, tertiary, and sometimes even the quaternary protein structures. So we may say that it is basically the sequence of amino acids which defines the shape of the protein. Now we'll just go through some of the very unique amino acids, and we are just going through these unique amino acids because in the upcoming videos where we will deal with the secondary and the tertiary structure of proteins, we'll see that presence of these amino acids, they define the further structural complexity or structural organization of the protein. Like glycine, it is the simplest amino acid in which the side chain, the R group, is a simple hydrogen atom. So this glycine, because of its minimal side chain, it allows tighter packaging of amino acids. And in this manner, it not only provides conformational flexibility because the two bonds, one by which the amino group is connected to the alpha carbon, and the other by which the carboxyl group is connected to this alpha carbon. These two bonds, they can rotate, so it permits conformational flexibility to the glycine molecule. And because of the tighter packaging, because the R group is very small, it is not a bulky group, various twists and turns, they are permitted in the polypeptide chain at the point where this glycine is present. Now similarly, there's another amino acid, the proline. And the characteristic feature of this proline molecule is that a cyclic structure is formed by its side chain, that is by its R group, with the amine functional group. So this results in the cyclization, and because of this cyclization within this molecule, it is able to exist in cis conformation, or rather it exists in cis conformation and not in trans conformation, like we have seen the two R groups on two different sides of the polypeptide chain, that is the trans conformation. And cis conformation means in which the R group of the adjoining amino acids, they are on the same sides. So if they are on the same sides, they will facilitate the presence or the formation of turns within this polypeptide sequence. So, proline is the only amino acid that permits cis conformation in the primary structure of, uh, sorry, in the cis conformation in the structural organization of the protein molecule. Another amino acid, a unique amino acid, is methionine, and it is unique because it is the starting amino acid for almost all of the proteins that are known in nature. Similarly, the cysteines, they have the thiol functional groups, and these thiol functional groups present on cysteines, they form covalent disulfide bonds with other cysteine molecules or with other cysteine residues present in the protein molecule. This again leads to the modification or the structural organization of the polypeptide chains. So these are some of the very unique amino acids that contributes towards the structural organization of the protein molecule. So if we talk of the various levels of organization of a protein molecule, there's the simple polypeptide sequence that forms the primary structure or the 1D structure. It is basically the linear order of the amino acids which are linked to each other by peptide bonds. And this leads to the unique folding patterns that is characteristic for a specific protein that is responsible for the specific shape and hence the specific function of a given protein. So this is the 1D structure or the primary structure. The second is the 2D structure, that is this polypeptide sequence, it takes a particular conformation, and that conformation, it results by the formation of hydrogen bonds within the molecule, that is within this polypeptide sequence. So as a result of these intramolecular interactions, this backbone of the polypeptide chain, a polypeptide sequence, it takes a particular conformation, and this structure formed is known as the secondary structure. It is basically the flexibility of the two bonds, that is the one which binds the amino group and the other which binds the carboxyl group with the alpha carbon, that leads to these particular flexibility of this polypeptide chain, and this leads to the secondary structure or the 2D structure. After that, more than one types of secondary structures, because till this point it is an intramolecular interaction. Now at the level of 3D or the tertiary structure, they may be intermolecular interactions. That is, their interactions, this formation of bonds, they may be the covalent bonds or the ionic bonds between various side chains. Here, hydrogen bond was formed between the backbone of the polypeptide sequence, and at this tertiary structure level, there is bond formation between these various secondary structures formed from this polypeptide sequence, and these bonds, they may be ionic or the covalent bonds. So in this manner, the protein molecule, it takes its specific structure in which some of the amino acids they are present on the surface, whereas most of the other amino acids they are present within. They are not in contact with the immediate environment. And at certain proteins, this tertiary structure it may lead to the formation of quaternary structures in which more than one types of these tertiary structures or more than one type of polypeptides they contribute into the formation of a functional protein. So these will be the structural organizational levels, that is the secondary structure and the tertiary and quaternary structures that we will deal in the videos that will follow. Thank you.